I will welcome everyone. Thank you for joining us tonight for this virtual Raise the Bar event brought to you by the Friends of the University of Alberta. My name is Ken Regan. I'm a vice president of the Friends organization. And I would like to begin by saying that the Friends of the University of Alberta respectfully acknowledges that we are located on Treaty 6 territory and respects the histories, languages, and cultures of First Nations, Métis, Inuit, and all First Peoples of Canada, whose presence continues to enrich our vibrant community. Welcome again. In just a moment, I'll be introducing our special guest speaker for tonight's event, but first I want to give you a quick backgrounder on the Friends of the University and provide you with some important information about the format of tonight's presentation. So firstly, the Friends of the University of Alberta is a not-for-profit society that's been around since 1943 and whose purpose is simply to engage in activities that help to build strong relationships between the university and the community, hence things like Raise the Bar. However, the Friends also produce the annual Henry Marshall Torrey Lecture. We fund a number of student scholarships and awards, and also support important programs like U School, a program that brings primary and junior high students, many of whom who are from disadvantaged backgrounds, to the University of Alberta for a week of immersive learning on campus. And as Friends of the University, we also act as champions and ambassadors for the University of Alberta within the broader community. I should mention that membership in the Friends is free. And though we gratefully accept donations, we do not solicit donations from members. So if you think you might like to join us, we are always happy to welcome new members. And if you'd like to know more about us or like more information, please go to www.friendsoftheuofa.ca and you will find everything that you need there to know more about us and the work that we do and the events that we put on. Okay, real quick, the program tonight will feature our speaker, followed by a question and answer session. The mics on your computers will be muted throughout the presentation, but if you would like to submit a question while Dr. Hurd is speaking, just do so using the chat function at any time. Our moderators will gather and relay your questions following the presentation. Once the presentation has finished, though, you can also ask a question in person by going to the reactions function and clicking on raise your hand. And one of our moderators will unmute you and you can then ask your question of Dr. Hurd directly. This evening's event will be recorded and posted on the Friends website at a later date. And lastly, for those of you who like to tweet, please do. And if you do, please include our Twitter handle, which is simply @FriendsOfUofA. Okay, now to the good stuff. Our guest speaker this evening is Dr. Christopher Hurd. He is a recipient of the 2013 Faculty of Science Research Award, an elected fellow of the Meteoritical Society. He is also principal director of the Institute for Space Science, Exploration, and Technology, which is a multidisciplinary virtual institute at the University of Alberta. And he is the creator and head of the University of Alberta's Meteorite Curation Lab, which is home to the largest university-based collection of meteorites in Canada and the world's first cold meteorite curation facility. And I don't think it would embarrass him by saying that he is one of the foremost experts on meteorites and I think Mars, probably in the known universe. But perhaps most pertinent to the context of tonight's discussion, he is an internationally recognized expert in the geology of Mars from studies of Martian meteorites, and he is a member of the current NASA Mars 2020-2021 Perseverance Rover mission as a participating scientist and expert in return sample science. So we are extremely fortunate tonight to have Chris join us to shed light on this latest NASA mission. and to provide his unique insights into what's up on Mars. So ladies and gentlemen, without further ado, Dr. Chris Hurd.
Hi, everyone. Thanks, Ken, for the introduction, and thanks for having me this evening. It's a pleasure to be here in a virtual sense. I'm going to share my screen. I've got some slides to share with you this evening. Martian delights and insights from NASA's Mars 2020 Perseverance rover mission. So what I'll do over the next few minutes is just give you an overview of the mission itself, what it's capable of, why we've sent this rover to Mars, what we hope to learn from it, and the long-term legacy that this mission is meant to have in terms of returning samples from the red planet. One of the biggest questions about Mars is what happened. At some point, based on previous missions, both orbiting missions and landed missions, on information from those missions, we understand that Mars may have looked something like what you see on the right-hand side of the screen. It may or may not have had a global ocean in the northern hemisphere, but it certainly had liquid water stable at the surface sometime in its ancient history, probably between 3.5 and 4 billion years ago. But today, it's essentially a lifeless desert. Then it had liquid water stable at the surface, it had a thicker atmosphere. Today, liquid water is not stable at the surface. It has a thinner atmosphere. It's only 1/100th the pressure of the atmosphere of the Earth. It's very dry, very dusty, and it has no magnetic field to protect it from radiation from space, from the solar wind. And so the surface is bathed in radiation. That was not the case, we think, 3.5 to 4 billion years ago, when there was an active, what we call geodynamo or magnetic field, as well as having these features that we see at the surface. So one of the biggest fundamental questions in planetary science is, why did Mars take this turn for the worst, if you will, somewhere around 3.5 billion years ago, to turn into this lifeless, dry, rusty desert, whereas the Earth, which is very similar, of course, is full of life. And then, of course, going along with that is the conditions we think were right for life back then on Mars. So what happened? So there used to be liquid water. So the big question is, did Mars ever have life? Well, if we look at the history of biology on the Earth, in this sort of sweeping view, we have a lot of single-celled organisms over the first really 2.5 to 3 or more billion years of Earth history. And then the first familiar fossils don't show up until about 600 or 500 million years ago, the first sort of familiar fossils. The time that we're talking about for Mars is around 3 to 4 billion years ago. So on the Earth, we had single-celled organisms that were capable of photosynthesis, that is taking advantage of sunlight for energy, for metabolism. So when we're looking for life on Mars, we're probably not looking for fish or plants or these classic fossils that are less than about 500 million years old, like we see on the Earth, because we think that that type of evolution takes a lot of time, literally billions of years. And Mars didn't have that time. That being said, we have microbial biosignatures, the biosignature being a general term for something that indicates life of a microbial type, starting 3.6 billion years ago, that's in the rock record on the Earth. So the question then is, what do we look for? Well, the answer is probably something like this. And on the right, we have what's called a stromatolite, which is essentially a layering in the rock record that we think is directly associated with cyanobacteria living close to the surface, the water sediment interface, and building up layers as sort of it would get swamped with some sand, and then the bacteria would have to kind of sort of make their way up towards the light within the oceans. So to understand the possibilities for life on Mars, we need to fulfill 4 ambitious goals, and these are the goals for the Perseverance rover mission. The first is good old geology, because you need geology to understand the context, you need geology to understand what rocks you're looking at and whether the environment that's preserved in those rocks is an environment that may have been habitable, what we call habitable. In other words, if life as we know it existed there, it would have thrived. The second, of course, is astrobiology, which is the study of something we haven't discovered yet, which is life outside of the Earth. That is what astrobiology is. It's using the principles that we have for how life may have arisen on the Earth and applying them to other places and trying to understand whether life existed or could have existed or did actually exist. And the third objective that goes hand in hand with the other two is actually to collect samples and bring them back to the Earth. There's a fourth objective that relates to human life, that is to actually prepare for sending humans to Mars sometime in the future. So let's look at our rover. This is going to be a brief overview. I could take questions about the different instruments afterwards, but just in the interest of time, I'll just summarize them here. It's the most capable rover ever sent to Mars, but of course, NASA wouldn't spend $2.6 billion for something or anything less than the most capable rover to ever be sent to Mars. And what you can see here, it's about the size of a small SUV, 6 wheels, to be able to traverse whatever type of terrain we may come across. It has a mast here in the middle, which has, as part of one of the components of the weather station, which takes things like temperature variations throughout the day and night, wind direction, relative humidity, that sort of thing. At the top of the mast, we have two very fundamental instruments that are including Mastcam-Z, which are panoramic stereo cameras that can zoom in or out, can look up close at rocks that are at our feet, basically, by the wheels, or out to large distances. And we already have, as I'll show you in a few minutes, we already have images from 2.5 kilometers away that are incredibly crisp. And then we have SuperCam, which is a combination, basically telescope, so we can actually get even as good, if not better, long distance images or up close images of rocks. And for rocks that are within about 7 meters distance, we can actually fire a laser at the rock and vaporize the rock and tell what the rock is made out of, both the elements and the minerals that are in the rock. So it's basically a laser gun for science. And then we also have within the rover itself, we have a RIMFAX, which is a subsurface radar provided by partners in Norway, which will allow us to probe the surface beneath the rover as we drive along, up to 10 to 15 meters depth. And then we have MOXIE, which is an experiment that brings in carbon dioxide, because the atmosphere of Mars is 96% carbon dioxide, and there's no oxygen to speak of. And it produces oxygen as a test bed for what we might need for future human explorers. And then the real business end of this mission is along at the end of this really beefy arm that you see here, which has two instruments that are kind of on opposite sides to each other. One's called Pixel, which is a way, when it goes up close to the rock, it allows us to tell, it fires X-rays at the rock, allows us to say what the elements are in the rock. And Sherlock, which allows us to look for mineral signatures, as well as even signatures for organic matter using ultraviolet. And then going along with Sherlock is Watson, which is a camera, which is basically like a hand lens, allows us to see really up close to the rocks. And then the cylinder thing in the middle here is what allows us to collect samples. So I'll talk about that later. Now, in addition to, from a technology demonstration side of things, I mentioned MOXIE, but we also are an interplanetary aircraft carrier because we have delivered a small helicopter to the surface. This is the helicopter known as Ingenuity. You can see it strapped here to the belly of the rover, and a animation of us essentially releasing it, the legs coming down, and then we drove about 5 meters away and then took this image. And it's a dual, this is a selfie taken with the Watson camera on the end of the arm looking back at the rover and at Ingenuity. And then here's a closer up view of Ingenuity. You can already see dust on the surface because Mars is a dusty place, but it is solar powered and it has 2 rotors that spin in opposite directions. And this technology demonstration is the first powered flight on another planet. But I'm kind of getting ahead of myself because let's talk about how we got here in the first place. The timeline for the mission was to select the landing site, to basically narrow down to the top 3 and then recommend the top, in order, the top 3. with Jezero crater, which is where we ended up being the top one. That was selected in a workshop in November 2018. I was fortunate enough to attend that. The rover had been designed and was being built in the interim, and then it was launched on July 30th of last year. It cruised to Mars. Oh, the launch was quite spectacular, although we didn't get to go because of COVID travel restrictions. But for those that were there, including this fellow who took a photo from about 5,000 feet over Cocoa Beach, you can see this arc into the sky, which must have been really quite something to behold. And then it cruised for 6 months and about 20 days, and then landed on Mars on February 18th, 2021. And now we're in surface operations. It was a 471 million kilometer trip. It was launched when Earth and Mars were close to each other. And then because of how orbits work, we basically had to spend those 6.5 months going 471 million kilometers to catch up to Mars. The actual landing event was really quite something. It's known as the 7 minutes of terror. It's called EDL for entry, descent, and landing. And I want to walk you through this. I'm going to show you a video that was released by NASA a few days after the landing. But on the upper left here, you see the cruise stage, which is basically that part of, so the rover is encapsulated inside this part, this capsule, with the back shell and the heat shield sort of sandwiched between those, and the cruise stage is attached to that. And the cruise stage is what allowed us to make corrections to the orbit all the way along from Earth to Mars, to make sure we're coming right down, threading the needle right into the exact position entering into the Martian atmosphere that we needed to. So the cruise stage separated, and then we have atmospheric entry. And at that point, the heating starts on the heat shield, of course, as it starts to enter the Martian atmosphere, and it starts to decelerate, of course, because of that friction. And then at some point it gets going, it slows down enough that the parachute can deploy, which is still deploying at supersonic speeds. Had to be specially designed to work in the thin Martian atmosphere. So it deployed, and then the heat shield could separate. And at that point, the rover could get radar lock on the surface of Mars in order to determine how high it was, what its altitude was. And then, and this is the most amazing technological event of this mission, it entered what's called terrain relative navigation. So what it did is it had maps loaded on board the spacecraft of the landing area where mission scientists had already determined, okay, this is an area, we knew we could get it within a particular ellipse, within the landing area, the Jezero crater. But within that landing ellipse, there are areas that are dangerous to land in, like it might be too rocky, so there might be a high standing boulder, or there might be sand dunes, which we can never climb out of, that sort of thing. So there was this hazard map essentially on board the spacecraft. And so the spacecraft, using images looking down at the surface, was registering what features on the surface and registering where it was relative to those maps. Then the back shell would separate, and then we enter what's called power descent, which is this descent stage, which is this thing with 4 rockets that allowed it to then divert laterally into a safe landing zone. And then it did what's called the sky crane maneuver, which is where the descent stage would lower the rover down on these tethers, set it down gently on the surface, and then fly away and crash. And all of this took place in about 7 minutes. But of course, because of the time delay in signals going from Earth to Mars and back again, the time delay one way was 11 minutes at this time. So there's no way anybody could be sitting with a joystick on the Earth or anything controlling any of this. It all had to be done automatically by the software on board. In addition, for the first time, all of this was documented with cameras that were on different parts, like on the back shell, on the bottom of the rover, on the top of the rover, looking up on the sky crane, etc. So what you'll see next is a video that documents all this. You'll hear the actual call of the person of Swati Mohan, the guidance and controls operations lead, calling the different, as we're getting the signal back, calling the different stages. But you'll see interspersed with this, you'll see the video from the different stages, and along the bottom is kind of a guide as to what part of the event was was happening at each time. I'm going to make sure I can share my computer sound. Okay, I'm going to go ahead and play this. And oh, the other thing I want to do is I will copy the video URL into the chat. In case this doesn't work, it is choppy because of a connection or whatever, you can watch it later on your own. Okay, I'm going to just let it play and I'll shut up.
We are starting the straighten up and fly right maneuver, where the spacecraft will jettison the entry balance masses in preparation for parachute deploy and to roll over to give the radar a better look at the ground.
Indicate chute deploy.
The navigation has confirmed that the parachute has deployed, and we are seeing significant deceleration in the velocity. Our current velocity is 440 meters per second at an altitude of about 12 kilometers from the surface of Mars.
Heat shield set.
Perseverance is now slowed to subsonic speeds, and the heat shield has been separated. This allows both the radar and the cameras to get their first look at the surface. Current velocity is 145 meters per second at an altitude of about 9.5 kilometers above the surface.
Nav filter converge. Velocity solution 3.3 meters per second. Altitude 7.4 kilometers.
Now has radar lock on the ground. Current velocity is about 100 meters per second, 6.6 kilometers above the surface of Mars. Perseverance is continuing to descend on the parachute. We are coming up on the initialization of terrain relative navigation and subsequently the priming of the landing engines. Our current velocity is about 90 meters per second at an altitude of 4.2 kilometers.
LVS valid.
We have confirmation that the lander vision system has produced a valid solution and part of terrain relative navigation.
Priming. TBA is nominal.
We have priming of the landing engines.
Back shell set.
Current velocity is 83 meters per second at about 2.6 kilometers from the surface of Mars. We have confirmation that the back shell has separated. We are currently performing the divert maneuver. Current velocity is about 75 meters per second at an altitude of about a kilometer off the surface of Mars.
Gear and safety Bravo.
We have completed our terrain relative navigation. Current speed is about 30 meters per second, altitude of about 300 meters off the surface of Mars. We have started our constant velocity accordion, which means we are conducting the sky crane, about to conduct the sky crane maneuver. Sky crane maneuver has started, about 20 meters off the surface.
We're getting signals from MRO.
Tango Delta.
Touchdown confirmed. Perseverance safely on the surface of Mars, ready to begin seeking the signs of past life.
I really don't get tired of showing that video. It's just absolutely phenomenal to go from something like 10 kilometers per second hitting the top of the atmosphere down to a gentle touchdown on the surface. And the fact that we could see everything, it just makes it so much more interesting. This was the first image here on the left shown after just about 40 seconds after, taken about 40 seconds after landing. And it's actually taken from what's called a hazard camera, which is under the belly of the rover. It's one of several cameras that allows us to drive autonomously eventually to around hazards, but it had a lens cap on it, but to protect it from dust during landing, but the lens cap is transparent. So it was able to take this image, which is the very first image taken. You can actually see the shadow of the rover on the surface, see rocks and soil. Very exciting, of course. Additional confirmation that the rover had landed successfully. Keep in mind, all of the video that I just showed you spliced together had not come down from the rover yet. One of the other first images that was taken was this one, which shows this plume off in the distance. This is actually the descent stage, that rocket-powered stage. Looks like something out of science fiction that flew away at the end, crashing about 600 meters away. So it actually caught the plume from that. That was about just under a minute after landing. And then, of course, the next few days we're checking things, initial checking things out, and then this is the first 360-degree panorama from the third day on the surface from Mastcam Z. So this is after we stood the mast up. You can see, interestingly, you can see the dust on the surface that's got kicked up. You can see where the rockets have blasted material away. These are these sort of blast zones here, kind of obliterated part of this dune. And it probably baked some of these rocks as well, but it exposed them. And then we can see everything off in the distance. We can actually see the crater rim the rim of the crater that we're in, these mountains in the distance. And then this line of mesas here is actually what's called a delta. And of course, the alien sky that we're under, this Martian sky. So let's talk about where we landed and why. We ended up in what's called Jezero crater, which is on the northwest corner of what's called the Isidis Impact Basin. It's in this part of Mars, if you're keeping track. And it is an interesting area, and it's a particularly interesting crater because it's about 45 kilometers across. And what we can see is that there is a valley called Naret Vivalis that leads into the crater, and another channel that leads out. So that by itself tells you that there was liquid water flowing into the crater and then out again on the other side at some point in its history. And some estimates put the depth of the water between 100 and 200 meters that filled up this crater at some point. This is the landing ellipse that I kind of referred to. This is where we knew we could put the rover down somewhere. And then within that, of course, we had that terrain relative navigation, so we could actually get down into a safe area. The most likely place was going to be right here, right off of the edge of the delta. I guess I should say that this delta right here, this pattern that you see here is from the sediment that was carried by this ancient river that was deposited by the river as it flowed into a standing body of water. The most likely place was going to be right here. We ended up over here. After landing, we named the landing site Octavia E. Butler after the female science fiction writer who has produced some seminal works. There's some science fiction fans on the team, myself included. We're pretty happy about this decision. So this is where we ended up. But the point is that we are at the bottom of a crater that may have looked like this at one point. This is an artist's impression of what the crater may have looked like, filled with water up to the brim. And obviously, the rocks that we explore in here may very well be recording an environment that was conducive to life. There's not a whole lot we can say, not a whole lot I can tell you so far, because we're still checking out all the instruments on the mission. And we really only have imagery to go with. We don't have a lot of chemical information. We have some from the laser gun, but when we landed, we could see that we have some rocks with holes in them, which we just call holy rocks for now. As you can see by the rover wheel here, we have light paver type rocks. We have darker rocks that stick up off in the distance, and then we have regolith, which is the name that we use to describe the loose material of the soil. Our little helicopter Ingenuity has done a fantastic job. It's been hugely successful. It's done all 5 of its flights now. So after landing, we did some exploring, a little bit of exploring. We moved off. We dropped certain parts under the belly pan cover, basically, and the cover for Ingenuity, which was strapped to the belly. Then we drove back close to where we landed, and we set Ingenuity down like I showed in that animation. And then we had to drive away ended up being about 75 meters away. And then we had to stay put for the better part of the last month in order to be able to be a relay for the Ingenuity mission on 5 different flights. This is from its 3rd flight. And the beauty of this little technology demonstration is it worked really well. It's gone as high as 10 meters now, I think it is. It's worked really well. The rotors have to go much faster than typical rotors on the Earth because of the atmosphere being so thin. It has a camera looking down, black and white, and has a side-looking camera, which is basically a cell phone camera, looking kind of at a 45. And this beautiful image right here was taken on the third flight. It had gone off, it had gone up and then over a ways and then came back and landed, and then taking pictures all along. And here you can see the ends of 2 of the feet, and you can see our rover off in the distance. And you can see the landing site. So if you look up close, here's a rover and the landing site where we landed with the blast zone and our rover tracks, we could see all of that. Now, in terms of the other technology demonstration part of this mission that has also been successful, on April 20th, I think it was, MOXIE did its first test and produced just under 6 grams of pure oxygen from the Martian atmosphere. And so that was successful, and actually did another test just the other day. We're waiting for the data to come down from actually, we've got the data down, and the MIT folks are working on that. So the 2 technology demonstrations have already demonstrated that we can do this, which is very exciting. And then of course, the other things are just getting these beautiful, beautiful images back, especially from Mastcam Z. So this is a panorama from the location where we were, actually where the rover was shown in that image from the helicopter, because that's what we call the Van Zyl overlook, where we were staying put in order to support the helicopter. And again, you can see, and this is a big panorama, you can download the full resolution. I mean, with a huge screen, you probably could just zoom right in. And you can see the rim of the crater off in the distance, these mountains. And then again, this mesa here in the sort of middle distance is our delta. And then this area right here is a place called Kodiak, which is kind of like a mesa on its own, which is probably was part of the delta at one point. And if you look at this, this is just astounding. We've looked at this several times of the day with different instruments, with both of the mast instruments. And what you can see here, which really remarkable, is you can see some flat line layers, and you can see some incline layers if you look closely here. And these are all consistent with a delta environment. That is where water is coming in from a river, flowing into a standing body of water. So it confirms what we suspect from orbit, and just is tantalizing because eventually we're going to end up driving, maybe not to this specific location, but we're going to end up driving up onto the delta to explore it more. Why are we going to do that? Well, because deltas, river deltas, are habitable environments. Here's an example from Alaska. What you might expect this might have looked like, maybe without the trees, on Mars 3.5 billion years ago. Key thing, though, is that we're going to go to these environments. We're going to go to the base of the delta, where we're hoping to find mudstones, ancient mud, which could preserve evidence of life. We are hoping to go to the edge of the ancient crater lake, maybe to where, because we know that the edges of lakes are good places for life to exist. But the thing is that whatever, and we're going to collect samples, but we need to be able to bring samples back to really answer that question of whether life ever existed there. And so the other part, of the part that's I'm most interested in, and because of my role in the mission, is within the belly, we have a sample caching system, and it's designed to the coring tube, basically, or the coring system is on the end of the arm. It's this big cylinder here. It's about, I think it's 85, so just shy of a meter long, like 2.5 feet, basically, in longest dimension. And this mates with this part of the rover here and grabs a coring bit of different, there's a number to choose from, but it'll grab a coring bit and a sample sleeve, and then it will touch on a surface of a rock and will core out that rock and go right into a sleeve. The sleeve will go back into the rover, where it'll get imaged to make sure we got something in the tube, and then it'll get sealed up and carried on board the rover. And this was just released the other day. This is a test that we did. It's not actually touching that interface that's up here, which is the sort of turret, which is where the coring bits and stuff are. But this shows you the business end of the coring system. So these 2 contact sensors will contact the surface and the coring and sort of stabilize it, and then the corer will come down and drill out a sample. And they look like this. So these are the sample tubes, and the sample that we hoped on the left there, and the sample we hope to get is about, I kind of liken it to kind of like a fat marker, like a, like a highlighter marker type thing in terms of the size. Each one would be between about 10 and 15 grams of material. What you see here on the right is this depot caching strategy. It's a bit of an older idea. What we're actually going to do is we're going to carry along the tubes, a certain number of them, up until certain points in the mission. Then we're going to put them down on the surface of Mars. These tubes sealed up individually on the surface of Mars in a grouping. And the current planning looks something like this. So we're actual landing sites over here. This is basically a geologic map that was developed before we landed, and based on a landing site closer to the delta. But the point is that the idea is this is basically what we're doing now. We're kind of redoing this now that we're on the ground and trying to determine where we want to collect samples most likely, because we know there are different units that we've mapped from orbit that are really interesting that we want samples of, and where are we going to get those samples from. So we might get a sample here, then we drive up on the delta, get another sample here, et cetera, and then another sample. And then we would get a certain number of samples, probably something like 20, and then we would put down 10 to 15 samples in an initial depot at the edge of the crater before heading outside Jezero crater to do more exploring and more sampling, to eventually go to a second place where we do a second depot. The whole idea now is that we would actually, by 3 years from now, we would be at this location right here on the crater rim, and we would put down a depot that might, and this is sort of like what you might put in the cache. You might have, this is what we envision in different samples that we might have. We would put them down in a cache, and then we would carry duplicates. So we would actually probably take 2 of each, or 2 of the greatest, we take greatest hits with us of the rocks inside of the crater, the ones that we think are the most significant, most important. We would set down one set, and then we'd take the other set along with us and keep going outside of Jezero crater, and then add more samples to it. And by the end of another 3 years, we would actually put down that final cache, which might have as many as 30 samples in it. So my role is indeed a return sample scientist. So I applied to NASA to be what's called a participating scientist. So in other words, I said, okay, I have expertise I think I can bring to the mission. They put a specific call out for people to participate, and NASA selected 10 people to participate from over 50 applicants. And I was one of those 10. I can't get NASA money, so the Canadian Space Agency said, okay, great job, we're going to give you money instead of NASA, which is really great, thanks to the Canadian Space Agency. And then we also added in a separate call, the European Space Agency added another 4 scientists. I think there's another 5th one going to be added to bring that part of the team to 15 scientists with specific expertise. And what is that expertise? It's basically what you do with samples in the lab after you collect them in the field, or after you receive them, after you collect them after they fall to the earth as meteorites or something like that. Another really interesting thing that happened was after I was selected, Ken Farley, the PI of the mission, called me and said, would you be willing to serve as a representative of return sample science on what's called the Project Science Group? And that's what this press release that was issued was about. And here I am listed as one of 2 representatives of return sample science, generally speaking, not just the people who were added to the team. Return sample science, which is a really major thrust of the mission. I am one of 2 representatives, along with my colleague Tanya Bosak from MIT. We are the representatives on what's called the Project Science Group, which is essentially the leadership council for the entire mission, which includes representatives from all the different instruments on the mission. So it's a huge honor, but it's also a dream come true for this guy right here. This is Chris at age 13. Really wanted to be there when samples come back from Mars. So this is a definite first step in that process. And so where we're at now is we've got the rover on the ground. We're going to start collecting samples. Our first sample we'll be able to collect after the 6th of July. We are doing more planning, like I mentioned, to sort of revamp that notional traverse out of the crater and figuring out where we want to most likely collect samples along the way. We're going to, by 2028, we're going to have the second cache done. If everything goes well and the rover stays healthy, we'll have the second cache done with the greatest hits from inside Jezero plus other stuff outside Jezero set down in a separate location outside Jezero. And then 2028, a fetch rover, which is being designed and built by the European Space Agency, will land at one of those two cache locations and collect up our samples, put them in a rocket, in a container on a rocket, and blast it off the surface of Mars, where the sample container will be captured by an orbiter, and which will turn around and bring it back to the Earth. Earliest return date 2031. So it's a little ways to wait, but at the same time, if we do our job well on this mission, we'll have at least 15, if not more like 30, a suite of distinct samples that could hold within them the mysteries of whether life ever existed on another planet other than the Earth. So it's going to be worth the wait, I think. And that's all I have for this evening. You can follow the mission at these locations. You can follow me at Space Rock Doc on Twitter. And of course, it's a NASA mission. And there's all kinds of phenomenal results coming down all of the time, press releases, but also the raw images. Everything that I've shown here is all publicly available. So all the raw images are automatically fed to the website all the time as they get downlinked, as they get downloaded from the mission itself. So I'll leave it there, and I'm happy to take questions. Thanks.
Great. I'll ask Tristan if you can bring me up again. So, Chris, thank you so much for that. It's absolutely fascinating and mind-boggling at the same time to see what's taking place in terms of space exploration in our lifetime. You know, I grew up, we were talking earlier, and I was saying to Chris that I remember Sputnik in 1959, and in my own lifetime, the fact that we have gone from the first Earth orbiter, unmanned Earth orbiter, to landing on the moon, and now a mission to Mars using technology and eventual human travel to Mars is just mind-boggling. We're going to take questions here in just a second, but if I may, I'd just like to ask a real quick question. Obviously, Chris, you have an intellectual and professional scientific interest and fascination in space and Mars and the cosmos, but I think it's something that is, I just think that it is primal. I think it is something that our species has wondered about and been fascinated about since we walked upright. And I'm just wondering, from your perspective, as someone who actually gets to work in the field of exploring space, why do you think that fascination is so integral to our being?
Yeah, it's sort of, you know, the early hominids moving out of Africa, moving into different environments, being able to adapt to those environments, exploring the Earth, wondering, it is that primal thing. It's sort of what's over that next mountain or what's in that next area. And you know, we've explored, with the exception of, you know, the deepest parts of the ocean, we've explored the Earth in great detail. When we can explore it sitting here, you know, with Google Earth, which is really kind of amazing just to think about that, to that development from Sputnik to that. I don't think people in 1959 could have even envisioned that. But yeah, so now we've had these tantalizing clues. And I think what's so fascinating about Mars is that we've had these tantalizing clues all along. And the whole question of whether life existed on Mars has flip-flopped in terms of in the popular culture, in the popular average person's mind, over the years from where it used to be that it was kind of accepted that there was a civilization on Mars, and then the first Mariner probes showed no, it's lifeless, and then another Mariner probe showed, well, actually there's these huge outflow channels that must have been water, and Carl Sagan saying, oh, there must have been life there. We might even see life when we send the Viking landers to the surface, and then the Viking landers showing, well, there isn't any life at the surface, it's sterile. And then as we explore it, as I alluded to at the beginning of my talk, we now really understand that the surface of Mars must have been saturated with all this water, and early in its history, and it's gone through this transition. So we can now start to look for life. It's not at the surface, we don't think. It's probably, if there is life today, it's well in the subsurface. But more importantly, the evidence is in the ancient past, most likely. But that question of whether, even if it's microbial, is a fundamental question. And it's one where if we find it, and we can say life exists on the next planet out from the sun, it must be abundant elsewhere in our solar system, but and all these other planets that we're finding, astronomers are finding all the time around other stars in other places in the galaxy and in the universe. So it really is a fundamental question. And if we don't find anything, I think that's equally interesting and exciting, because it tells us that there really is something unique to the Earth, to the conditions on the Earth that led to life here and not there.
Thank you for that. Let's do questions. I think, Tristan, are you okay if I just read these off? We'll take them kind of in the order they came in, Chris. And we had an email question actually before this evening from someone who asks, what kind of rocks are you most hoping to find on Mars, and why? And just for fun, what if you do find footprints of little green men?
The rocks that I'm hoping to find, so I've spent my career working on meteorites from Mars, because that 13-year-old kid said, I want to work on rocks from Mars, and until we bring samples back, the next best thing is going to be these meteorites. So I've been working on those. Those are igneous rocks. They're actually, I didn't get into it, but there's kind of a bias in how they're delivered from Mars. Something has to hit the surface of Mars and blast rocks off the surface. And there's kind of a bias towards those rocks that are more likely to make that trip or survive that process. And so they actually are mostly igneous rocks, sort of from lava flows, probably from Tharsis or Elysium, from those areas that have had volcanism more recently. And so they're much, much younger than the rocks that we're sitting on even right now, we think. I am still hoping to find rocks like that, igneous rocks like that, because we know so much about Mars from the igneous rocks in the Martian meteorite suite, that if we could find an igneous rock, especially if we can find it like in an outcrop, like not detached from, from like what we call in place, like what geologists really like to see. Like when you see a road cut, we've cut through that rock. You're not looking at boulders, you're looking at the rock face. We want to find that. If we can find that, an igneous rock like that, it's in this area is probably going to be 3.5, 4 billion years old. It'd be really great to get a sample of that and compare it to what we see in the meteorites, because we think they're going to give us very complementary information. If we see footprints from the little green men, we will need to carefully assess whether that is likely to be evidence of what we call bioturbation from organisms walking across the surface, and rule out all other possible mechanisms.
I'm glad you've got that covered. So another question, does the lack of magnetic field mean that Mars gets more spectacular aurora than we do on Earth? Or does the thin atmosphere mean they get all the radiation and none of the light show?
I think it's the latter. I think they get all the radiation and none of the light show. Yeah, in my understanding of how aurora work, yeah.
That was a question from Paul Childs. We have another couple of questions from Michael Mant. He says, I understand that not only are there organisms in hydrothermal vents, but also deep underground with different energy chemistry than most other known organisms. Will the NASA scientists be able to test for life that may be totally different from Earth? And the second question, it's likely impossible not to transport Earth organisms on space missions, for example, radiation resistant or in biofilm. Any comments on that? So the first one about being able to detect, test for life that may be totally different from Earth. And the second one, what about the potential to transport Earth organisms to the surface?
For the first question, we can really only test for life as we know it. We can certainly, it's a really good question. It's really at the heart of astrobiology. We know we have one example of life and how it works and the chemistry of life. So we can really only test for that. The hydrothermal organisms on the Earth are still based on the same chemistry of life, and they still use DNA or RNA. And so they are able to, so we could still potentially detect that. Now, we're not expecting to find DNA. What we're expecting to find is a chemical signature of life being there. And there's certain things like isotopes of oxygen or carbon, things like that, that we know where metabolism has been involved, it leaves a trace, it leaves a particular signature. And so that's, again, that sort of gets to what my point was about, we need those samples back in the lab. Our rover is capable as it is, cannot do that type of analysis. That's the type of analysis that needs to be done in labs on the Earth with the best possible mass spectrometers and all that. So we would be looking for, most likely for the chemical signatures of life as we know it. That being said, if life as we don't know it existed, it might leave a chemical signature that we could say, you know what, we can't explain this chemical signature by any other mechanism. Like there's no natural processes that we know of that could explain that particular chemical signature. The second question is a really interesting one and really important. It's what's called planetary protection. There's a whole other office at NASA that deals with this. And it's called forward contamination is bringing microorganisms from the Earth to Mars. And backward contamination is if there's something on Mars that is actually extant, like actually living, we want to make sure it doesn't contaminate the Earth. The second was really, really unlikely because of the radiation, although the samples, when they come back, will be going into a special sample receiving facility and will be fully characterized for any kind of extant life. The forward contamination is a big factor for the spacecraft. The whole spacecraft was carefully cleaned, and those tubes that the samples are going to go in are probably the cleanest things that we've ever sent into space, because we want to make sure there are absolutely no bugs and no dead bugs and no dead bug bodies in those tubes, or even as tiny as low a possible amount of any even just organic matter, organic compounds. So those have been extremely carefully cleaned and put in the spacecraft at actually the last possible minute. And so to prevent that forward contamination, because we don't want a false positive is what it comes down to. We don't want to spend all this money, bring the samples back, and then realize, hey, you know what, there's actually bacteria from Florida that actually are in this sample tube.
Right, bacteria from Florida on Mars. Go figure. So thank you for that. A question from Robin Owens, who says, and this is a great question, I think, why did Mars lose all of its water?
It's a big question. We think that a lot of the water ended up in the subsurface as solid. It froze out basically. So we think there's what we call a cryosphere. So there's water in the subsurface as ice. There's a paper that came out recently, actually by a grad student at Caltech, who's also a member of our team, Ava Scheller, who I think the gist of it is that there was enough reaction of that early liquid water with the minerals in the crust to bond with and form new minerals that took up the water. But we also may have lost some to space. But really, it's a drying out through a number of mechanisms, and the atmosphere, losing the atmosphere too, somehow, bleeding off into space, probably, especially the magnetosphere stopping could have helped with that, is probably a factor as well. So there's all these things that we're trying to figure out sort of the sequence of things that happen and why, and the sort of causality of it all.
Gord Gilroy asks, from your perspective, Chris, what is the value of putting a human on Mars? versus using machines to gather information?
You know what's really kind of amazing, and this is the first mission I've been involved in, and I think what's really, like I just described to you, the 7 minutes of terror, the phenomenal accomplishment of putting this huge rover down gently on the surface, flying a helicopter, all that stuff. We're driving around now. We still don't know what the rocks are on the surface, what they actually are. They're probably igneous or sedimentary. That's all we can say right now. We're getting some clues. We're getting some hints. It's true, we haven't commissioned all the instruments yet. We don't have everything on the arm working yet. And we're not able to, there's another tool that we have, instead of a coring device, we can actually abrade the surface to sort of clean it off. But if you put a human down, and you've been a human there for 80 days like we've been so far, we will have walked this entire area probably several times, have picked up rocks, broken them open, looked inside, determined what minerals are there, intuitively gone over to the other rocks that we see, we've on the team have identified as different type rocks, determine what they are, their relationships with each other. We would have done all of that intuitively. So there is as capable as these rovers are, there's an incredible amount that we can do as humans that is so intuitive that is nearly impossible to replicate with a rover. Well, a rover is extremely capable, and we can do all these analyses of chemistry and all that, which would be harder to do with a human. It'd be cool to have a laser gun on your belt that you could zap the rocks with and tell you what it is, right? And so there's all these things that the rover can do. But what you're doing is you're trading the time that it takes. You're trading something that doesn't need oxygen and food to survive with something that takes longer to do the things that humans intuitively do.
Robin Owens asks, what do you think of the putative evidence for microbial life in the atmosphere of Venus, evidenced by the spectral signature of phosphine?
I've sort of been following that debate peripherally. It's sort of like, it's a little bit like Mars extant life was in the 70s. Oh, there must be life there. No, there's not. And so I think the signature, the actual spectral signature, has been debated. And I think the most recent thing is that, oh, actually the signature is not there. It is an interesting idea. The idea is that Venus is this hellish place where life couldn't possibly exist at the surface, but up in the cloud deck where there's liquid water, it's still acidic water droplets, but it's at the right temperature, sort of more like room temperature, you could end up having bacteria survive. But yeah, it is one of those things. It falls in the category of astrobiology, and it is one of those things where you need sort of dedicated studies in order to really try to answer that question, whether it really does exist.
I think we're just about out of time, but I want to get in this last question from Andrew Greenshaw. And Andrew says, I think this is a great question to end on, Chris. He says, what would you say to the young Chris about the future of Mars exploration?
I'd say you're not going to believe it. Like it's in just a short amount of time, 30 years or so, have a bit of patience. It's going to happen, but we're actually going to have a rover on Mars that can collect samples and bring them back, and you'll have a good shot at studying them. I think I think that would be pretty exciting for 13-year-old me to hear.
No doubt. Well, I will say, as we wrap this up, I will thank you sincerely for doing this and for such a fascinating virtual exploration of the red planet. And someone complimented me on my use of my red glass tonight. So, Chris, thank you so much. You've been a great friend to the Friends of the University, and we really appreciate your insights and your support. So, a virtual round or a real round of applause for Dr. Chris Hurd.
Thanks so much, Ken. It is my pleasure. I do enjoy these talks, and you can tell I do get excited about it. And I don't see that excitement waning anytime soon either. It's quite something to be involved in. So.
That's
Thanks.
Great. Well, we certainly wish you well with the remainder of the mission and all of your future research and explorations. Before we go, I would also like to thank or to let you all know as well that on November 4th at 7 p.m., the Friends will be presenting the annual Henry Marshall Torrey Lecture featuring Professor Andy Knight, Professor of Political Science at the U of A, whose work includes research on social justice, exploitation of child soldiers, and global governance and terrorism, among many other things. And just this March, in fact, Dr. Knight was named a distinguished professor at the U of A. So mark that in your calendars, November 4th. Stay tuned for further information. Again, if you would like to know more about the upcoming Torrey Lecture or anything to do with the Friends of the University of Alberta, including membership and or joining our board of directors, please go to the website www.friendsoftheuofa.ca. I want to say thanks again to our speaker, Dr. Christopher Hurd, and also to all of you who joined us this evening. I hope you'll stay tuned for more Raise the Bar events, hopefully in person at a local bar or lounge where we can actually have a real drink. And I want to thank you for your participation, everyone. Thanks to our team, Luca, for our president, for technical management, and to Tristan Cruz and Andrea Sulima for being our moderators. My name is Ken Regan. Thank you so much for being with us, and have a great evening.